Introduction. Dr. Ashutosh Jani, now he's the head of clinical operations at Veeda Clinical Research, Gujarat, and where his expertise or his area of research include oversee the conduct of patient-based PK and clinical endpoint studies, proof of concept studies, and late-phase studies for ANDA, EMA, and DCJ submissions, and oversee their execution. An operational expert for new business and program or project delivery lifecycle, and who is accountable for overseas and supervision of projects and study teams to ensure the quality of deliverables on time and within budget. And earlier, he worked as a senior general manager clinical trials at Lambda Therapeutic Research, and before that, he was a head clinical trials of Acutest Research Laboratories, Ahmedabad. With this brief background, I invited Dr. Ashutosh Jani to deliver a talk. Yeah, thank you, Doctor.
Sure. Okay. Thank you, Som Sundar, for the introduction, and a very good morning to everyone. So I think you have been bombarded with a lot of biosimilar related information in the past. So I would continue to do that, and think that would help you in better understanding of the subject per se. So, okay. Are you able to see the screen or not yet?
Not yet.
Yeah. Okay. Okay, so I think you're able to see the screen, right?
Yes, we can see.
So as a small exercise, just to wake you all up, we'll have a small exercise to get a fair idea about who is there today or the meeting. I would say the con. So just yeah. So I think you'll be able to see us. You will see the screen, Kahoot, and this is the pin. So if you all can log in, and then we can probably have a start. Just wanted to know the people around. So if you can just go to kahoot.it and enter this pin. So yes, so thank you. Yeah, anyone else? It's a simple thing. You just have to go to www.kahoot.it. You could do it from your browser or a mobile browser, mobile app, whatever is convenient. Hello, good morning, Naveen. Thank you.
Good morning, sir.
Only two guys. Yeah, it's a simple thing. I don't think so there is any complexity involved. You just need to go to www.kahoot.it and enter this pin. I think people in the last day moved. Yes, guys, we're waiting for you. I don't think so we'll be able to start without you. We just don't want a monologue sort of a thing where I just go on and you guys are not attentive. So we just need your small attention. Everybody just go to www.kahoot.it and enter this PIN number. Hello. I think we are okay. We are at least 17, right? So there's only one who has just logged in. Okay, two. Is it that difficult? Thanks, Vaibhav. This won't allow me to start. The problem is that unless I have sufficient people on, it would not work. So request everyone to just log into Kahoot and okay. So we start off with just three guys here. So just wanted to know the audience. So you could just help me out with you could select the concern answer and we can take it up. Okay, only one. Okay. Okay, so both of them are graduate students. Good to know that. Okay, so let's start off with the presentation for today. Okay, so thank you everyone, and here we go with the presentation. So what we'll be just touching upon would be, anyway, you all are know what's a biosimilar is a very vast subject to discuss on. We'll be touching a very small aspect of the same with respect to the clinical efficacy studies, because that's a really important part of the evolution of biosimilars. That brings me to this story of this movie. I'm not sure if you have seen this. The name of the movie is Living Proof, and it's based on the true story. So, if anyone knows Herceptin, Herceptin is one of the biosimilar which is used for triple negative breast cancer. It's a bit, basically discovered for breast cancer. And this is a beautiful movie depicting onto the different problems which were encountered during the discovery and the clinical part per se, enrolling the females of breast cancer for this study. So if you get a time, just Google it out or YouTube or Google. It's definitely a movie which I would recommend. It's based on the discovery of Herceptin. Now, as we've seen that the whole week you've been bombarded with lots of biosimilar related talk. So that brings me to the history. So how these things originally started. So if it goes back nearly about 1980s, where recombinant DNA, through the recombinant DNA, the insulin was prepared. So that was regarded as a breakthrough achievement during the 80s. And gradually things progressed in the next decade of 90s, where then there were some guidelines with respect to comparability guidelines, because insulin was not a small molecule like any other drug. It was basically a protein. And then there was a gradual development of comparative guidelines. In the next decade, in about 2000, year about 2000, there were small improvement which led to the development of the guidelines from the Europe and the WHO organization, the World Health Organization. So they gradually came up with the biosimilar guidelines, and nearly by 2015 and today where we are at 2021, on the verge of entering 2022, there has been a drastic improvement in the way the biosimilars has been progressing. So that's what typical biosimilars market, or that's how important it is. Just to briefly give you a fair idea of what biosimilars are. So we have different in India we have different mangoes available in different part of the country. So here we have in part of where I live, the Gujarat, we have the Kesar mangoes. In the Maharashtra region, we have the Ratnagiri, Alfonso mangoes, and so on and so forth. So it's the same fruit per se, mango, almost similar taste and flavor, sort of. And that's typically different variety, but it's basically called a mango. On the same lines, there are cheese. We have Parmesan cheese and goat milk cheese and so on and so forth. So there are a variety of cheese which are available. Same way they are processed as we process any protein molecule. They are also proteins. And we have different types of cheese, but basically they are cheese. They are a good source of protein, etc., etc. On the same lines, we have biosimilars. So they are almost the same, like the one which is the innovator molecule which is available in the market, and they produce almost the similar actions. So as an analog, we could compare it that there is a lock, there's a problem, there is a disease. Now you have an original key which helps you in opening this lock, and then what you try to do is you try to duplicate the original key. Now, even if you try to duplicate the original key, you will not be able to completely duplicate the original key as it is, because that's the original. The main key teeth lines where which are responsible for opening of the lock, they may be quite similar. The rest, there could be small change or difference in the arrangement of the teeth on the key. So on the same lines, we have biosimilars. So they are typically and duplicate key opening the same lock which has been opened by any innovator biologic. Now, if you consider, even if you start off with the same ingredient, I mean, say everything is the same as the innovator, and try to make a copy of that, you can never duplicate the same thing. So that's even true with respect to biologics. I mean, say in our case, where it's the same sperm and the same ovum which gets fertilized, but when we have twins, they may be identical twins, but despite them being identical, their their fingerprints would be different. On the same lines, even if you start off with the same material, the process would be slightly different, and that would give you to a different output altogether. And that's how we differentiate typical biologics and a biosimilar. So you are almost there, but still you are different. Now, there are different ways in which you get to, if you're a biologic, and say the first person on the original key, if we consider that way, then you need to do a battery of studies, right from the clinical pharmacology, non-clinical. So there are loads of clinical studies, clinical pharmacology, non-clinical, and analytical studies. But if you are a follow-on biologics or a biosimilar, then you don't need to do those battery of clinical studies. So the clinical studies portion gets reduced, as you could see in the diagram. And there's more focus on to the analytical, non-clinical aspect, and very less onto the clinical aspect when we try to compare the two drugs. This sort of summarizes the innovator development. I would say any company who is just starting off with developing biologic, they need to start. The process goes nearly about 20, 25 years long process, right from discovery, validation of the target, to produce the cell lines, finalize the process, you get a fair understanding about the products, and then you monitor, scale up, and then you do the clinical studies, the non-clinical studies, clinical studies, and then other additional indication studies. So it takes a long time for an innovator to get into the market. However, for a biosimilar development, you have just need the typical cell line process development characterization, the analytical characterization, and the specifications, and very few non-clinical studies and very few clinical studies, and you get a pre-approval based on the safety data which you produce. So this is a typical comparison of an innovator development versus a biosimilar development. So that's quick for sure as compared to the innovator development. The time required is less. Now, why is this a hot thing now, and why are you bombarded with so many things with respect to biosimilar in the last week per se? So the major factor being that the focus is more onto the patent expiry. So if we consider 2022 as a year, we have a pretty good number of drugs. I'm just talking of the US FDA. And there are pretty good drugs like Bevacizumab. They are facing almost on the verge of patent expiry. So that would give an opportunity for other people to get onto these markets. And it's estimated that there's a huge market out there if we consider just the key areas like the US, EU, Japan, and Canada, the key players. There's a huge market out there. And almost 17% of the spend is allocated to biologics at this point of time. If we consider the spend is gradually increasing, at this point it's 17%, and the sales is definitely increasing year on year. So that's the thing. These typically summarizes the things which make biosimilar really a hot topic now. And just adding to why they are hot, because just few years down the line, if we consider, then we just had biologics like biosimilars like say erythropoietin or GCSF immunology. Those were just the indications which were targeted in the last 5 years. At this point of time, if we consider 2020, we have biosimilars which are targeting various other therapeutic areas like say diabetes for the insulin, erythropoietin, anticoagulants. Immunology and oncology definitely remains the major player. So there's a huge market out there which is growing exponentially. Which is 75% of biosimilar medicine sales are just on oncology and immunology. Coming on to the spending of the first biosimilar availability. So we start with Herceptin, and that's trastuzumab, and then we go on to the latest one, Humira, which is adalimumab. You could see that there's a huge spending which is happening from various countries out there, and the various availability of these biosimilars over a period of time in various countries. So if we consider the pipeline, as we were just mentioning, so these have a really huge pipeline, and there are certain molecules which are either in file stage, so they have filed for the approval, or they have already been approved, and certain were already marketed at this point of time. And if we consider in the coming years, there are definitely pretty much a number of biosimilars which are expected to enter into the market in the next 5 to 10 years. I'm not sure if you are aware how they are named, because as I mentioned, they are quite similar. I mean, it's very difficult to differentiate between the two. Only a minor change in the structure. A few amino acid sequencing is changed, and that's how the biologics are prepared. So if we go with the normal nomenclature, they are typically, when it comes to biosimilars, they are nomenclature or given a name. Typically for the normal drugs, as we have rosuvastatin, we call it as Crestor or Rosuvac or whatever. I mean, it's a different brand names, but the drug is still rosuvastatin. But that's not the case with an biosimilar. Like say if you have Remicade, it's Infliximab. But the different Infliximabs which are there, if it comes from the first company, second company, third company, how will you try to differentiate the Infliximab? Because they are typically different, as we've seen. So there's a way to nomenclature them, and based on the core biological name, that's Infliximab. Those are the code name. And then there are 4 letters, WXYZ, which would be given based on the company and which it's working on. So coming on to the clinical studies and the aspect. So if we consider the clinical efficacy studies for these biologics which have been conducted, there is a really important aspect with respect to design, because that's really an important criteria wherever we are evaluating any biosimilar. So first we need to design the study per se, just get a fair idea about the time, how much time we have, how much would the treatment period, the resources available, and it should not be that it's that long that by that time, I'm to say, the market doesn't there or market gets saturated. So you need to factor in all these aspects while designing the study. So even if the study is not well designed, I'm say it'll be sheer waste of time and money, and you will reach nowhere with respect to your conclusion. So, if we consider the key design considerations, so the main would be we'll see these things in detail in the coming slides. But the key design considerations would be more onto the indication. Like say we have adalimumab, then adalimumab would be used or it's indicated in various indications. Now, which indication is what we are targeting? Or if we considered an indication, that would definitely bring you to the type of patient population. So what type of patients would be looking at? Say if you're looking at patients with say rheumatoid arthritis, then how much should be the degree of rheumatoid arthritis? It should be, I mean to say, how many joints should be affected, how many tendons should be affected. Those things would be also an important area to be considered during the designing of the study. The treatment regime, so whether the route of administration is it I would say intramuscular, subcutaneous. So whatever the injectable route of administration is, would also be an important criteria in designing the study. Apart from that, the most important would definitely be an endpoint. So what you'll be looking for to get an idea about whether the drug is working or it's not working. So that would be your typical endpoint, which you'll try to see to get an idea and the effect of this drug, the efficacy part. The study duration also plays a vital important role in the designing, because a study can be as short as say 4 weeks, 6 weeks, or it could be as long as 24 weeks. So that we need to factor in that how what is the effect of the administration of these drugs over a period of time, whether you get the effect which you could see within say 4 weeks, 6 weeks, or it takes more than 20, 24 weeks to get an idea about the effect of the drug. And there are comparative designs also. So if we try to break these things per se, we just try to break these factors which are affecting the study design into four key factors, and we consider that as patient-related factors. It could be related to the disease, either it could be the endpoint or certain design factors. So these are sort of summary of what are the factors which would be governing the typical efficacy study and its design. I think you are aware about randomized matched group design or stratification. So what happens is, like say you have all different types of people available at your disposal for conduct of the study. Now it could be short, long, thin, healthy, obese, whatever. So there are a variety of patient population which is available, and you need to ensure that you have a equal distribution or quite a similar distribution in both the groups. So it should not be that all the thin, all the short people are in one group and all the long or all the obese people are in the other group, to get a fair idea about the effect of the drug. Now, in order to do this, we need to rely on randomization. Randomization is a process by which we'll be randomly allocating them to either of the treatment arm. It could be either group A or group B. But we'll also like to stratify them. So as you could see in the picture, there is a similar mix of people in both the groups. Now, this could be possible only with the stratification. So you stratify that if you have one patient which is long in one group, there will be a long in another group. A short in one group, the next short would go in the other group. and that's how you stratify them. And this randomization is typically a gold standard when we come to clinical trials. And definitely it is essential that we randomize these onto the standard the standard procedure way. So if we come to this, the same example which we were seeing on the adalimumab sort of thing, what happens is, let's say there are various companies which are into the market developing this drug, and what say, like say they have put up is in their, I'm just trying to summarize them on all the studies which they have conducted. So there is a drug methotrexate which is used along with this in the treatment. Now, what happens is previous biological therapy. So Amgen is doing the study where they are allowing patients who have already been exposed to previous biological therapy. So if they have been treated with any other biologic in the past, they can still be part of this study. And similarly is the case with Sandoz or Mylan. But however, Coherus is doing the study, so they don't want previous anti-TNF alpha exposure. So they are just looking at patients who have been exposed to any biologics in the past. Now, what happens because of that? We'll be seeing those in the next slide. But then you also need to ensure the blinding or the masking of the treatment, because there are various factors which play an important role in these. One of them is the ethics, because you're double masking the procedure, so it's an undue risk to the patient. No one is aware of what treatment the patient is getting, and practically at times it's really difficult to mask the treatment, because one could be a pre-filled syringe, other could be a vial, and so on and so forth. One could be a plain color, the other could be maybe a white crystalline sort of formulation. So those things would be different, and there are chances that you may end up compromising with respect to the blinding of the product. So, as I was just mentioning, Humira was one of them, and it is marketed typically as just which one? Humira is typically marketed as a pre-filled syringe, and that can be easily recognized. So the study drug adalimumab, if we try to bring that into the market, and if it's in the vial form, then it's typically difficult to blind these formulations. As we are seeing, one of the other criteria was with respect to stratification. So adalimumab for that is indicated in plague psoriasis, rheumatoid arthritis. These are the two indications in which they have done various studies on. And if we consider plague psoriasis, then there need to be a PASI score which needs to be enrolled for the study, and the treatment duration is nearly 16 weeks. And it has been seen that efficacy is decreased mainly in patients with body weight more than 90 kg or with mass index or BMI more than 30. So if you're doing study on adalimumab for plague psoriasis, you would not like to have obese patients or patients with more than 90 kg of weight or with BMI more than 30, because that is or tend to affect your efficacy of the drug. Similarly, when we come to rheumatoid arthritis, so we would like to stratify the patient. As we've seen in the previous slide, that you can have one short, one long, tall, thin, everyone should be equally distributed. So if this happens that there's no stratified randomization and we have all people with high weight in one group and low weight in other group, the study results may be haywire. I mean, say there could be uh, a difference in the results. So that's the reason we like to stratify them for plaque psoriasis. In the same case for rheumatoid arthritis, also the stratification is based on the disease activity. So there is a DAS score or disease activity score. Now, this would be helping you in getting a fair idea about the involvement of joints, which type of joints are involved, whether there's involvement of soft joints, major joints. And based on that, you determine the disease of rheumatoid arthritis. So you would like to score them and stratify them equally in both the treatment arms. So, as you are saying that adalimumab is being used in various indications like rheumatoid arthritis, psoriatic arthritis, ankylosing spondylitis, Crohn's disease, ulcerative colitis, and plaque psoriasis. So there are these various indications in which adalimumab is being treated as a TNF inhibitor. And what they try to see in different indications is you could see it on the right hand side that there are different efficacy analysis which has been uh, so you could see that there are different efficacy endpoints like ACR20 is regarded as one of the efficacy endpoint. There are concomitant medications like methotrexate, as we saw. They are allowing, some of them are not allowing. The treatment duration is different. Like say one of them is 24 weeks. The other in ankylosing spondylitis, it's just 12 weeks, and Crohn's disease is as low as 4 weeks. Because it's proven that if you're trying to go for Crohn's disease, the effect would you'll be able to see the effect within 4 weeks of time. But in case of rheumatoid arthritis, it takes a long period, maybe up to 52 weeks, to get a fair idea about the effect of the drug. So these factors would typically be helping you in characterizing your design for the particular study. So, as I mentioned, patients were one of the factors with respect to the study design. And the next, as we were seeing, is the disease activity. So, what is the amount of disease or how severity of the disease is? So, if we compare all the adalimumab studies which are ongoing, just then Amgen is doing Sandoz, Mylan, Coeurus. So they all are doing these studies. Then the disease activity is like say they are enrolling only patients with more than 12. That's common for all. Body surface area is considered as 10 for all, and the SPGA is nearly 3. On the same lines, if we consider the disease activity here, they are looking at joints, I mean, say swollen joints because of rheumatoid arthritis or the tender joints. Their number is nearly more than 6 to be part of the study. When it comes to other clientele, they are also looking at CRP, C-reactive protein, as one of the factor which could be used for enrollment of the study. And for Samsung, they are also considering the erythrocyte sedimentation rate and C-reactive protein. Coming to previous biologics therapy, they are allowing previous biologics therapy. However, Pfizer and Samsung are not allowing previous biologics to be part of the study. Now, how these affects is like say if you are allowing permitting either previously exposed biologics, then what happens is like say if you go to countries like US, Europe, Japan, and Canada, where there is already patients have been getting biologics treatment in the available. So there would be that you may get patients who have already been treated But then there are certain countries where the biologics are not there. So if you want to do, like say Pfizer or Samsung wants to do a study where they are not permitting prior exposure, then they may need to go to other countries, like say Southeast Asian countries, where there would be no biologics which would be available, or there will be very few patients who could be on biologics. So then you get an easy recruitment year. So these factors are definitely important, and they are typically considered as important for the designing of the efficacy studies. These are the various head-to-head comparison or various things which we need from the reference drug as well as the biosimilar. So you need to have a match with respect to the structural, biological, non-clinical, and PK/PD aspect. Those should be a match. The sensitivity could be different with respect to the indication 1, indication 2, or indication different indication. Like say, it could be effective in rheumatoid arthritis, but it may not be effective in plaque psoriasis. So if we consider clinical efficacy studies for adalimumab, so what they do is they have this various indications available, like say plaque psoriasis or rheumatoid arthritis. But then the company would like to target the indication which is either easy to enroll, or very less differences, or highly sensitive. So the principle that sensitivity for detecting the small difference between the agent is the highest, that particular indication is what we would like to target. So if you are giving adalimumab to a person who is suffering from, say, rheumatoid arthritis, and the same different drug you are giving to the same another patient of rheumatoid arthritis, you should be able to see the difference sharply. So that's what is what we are looking for, detecting the difference between the two agents. Now, there is less difference when it comes to rheumatoid arthritis, but when it's come to plaque psoriasis, you could visibly see the difference very quickly. So if you are targeting in plaque psoriasis, your sample size could be less, but however you will get, so getting the patients of plaque psoriasis could be difficult. However, when we come to rheumatoid arthritis, you could easily get patients of rheumatoid arthritis, but you may need more and more patients for the study of rheumatoid arthritis. Coming on to the extrapolation of indication. So the patients are, I mean, say if we target one particular disease indication, like for example, we're targeting rheumatoid arthritis. So then if your drug is effective in rheumatoid arthritis, you may or may not get approval for other indications of rheumatoid arthritis. You may not get the approval for indications of plaque psoriasis or any other indication. So yeah, this is this sort of an extrapolation of indication summary, like Europe, Health Canada, Australia, Japan. So these are the various indications: rheumatoid arthritis, plaque psoriasis, psoriatic arthritis, or ulcerative colitis, Crohn's disease. So these, if you are getting an approval for one indication, and then you can extrapolate it to other indications in Europe, that's possible. But when you come to Health Canada, you can extrapolate to few of the indication, but not possible to Crohn's disease. Now, Europe's Crohn's and colitis organization, they emphasize the difficulty to extrapolate across indication. So they have raised a concern, and that's the reason you may not be able to extrapolate it to the other indication. And you may have to do a separate study for Crohn's disease to prove that the drug is effective in Crohn's disease. Same is the case with, like, say, if you have it in Japan, you get for rheumatoid arthritis, you may extrapolate to this, but you may not extrapolate to other indications. So every regulatory has its own way of functioning and extrapolating of the data to other indications. So as we were just mentioning, so there should be a proper justification for extrapolation to the other indication. Apart from extrapolation, the other important thing is interchangeability. Like say, if you are on formulation A uh, in the initial part, can that be switched to a drug B, which is a biosimilar? So that is interchangeability. And then vice versa. If you are on B, can you put on A? So that is interchange. Instead of A, you give B. Instead of B, you give A, as we do it normally for small molecules. If you go to the medical store, you could take paracetamol of company A or paracetamol of company B. Either way is fine. The next is substitution. So if you are substituting, say instead of A, you can give B, but instead of B, you cannot give A. So that is substitution. Interchangeability is instead of A, B, B, A, you can interchange between the same. Substitution is only you can substitute A with B, but you cannot substitute B with A. And there is something like switching. So the decision to switch, I mean, say if the patient is either on one treatment, switching them over to another drug treatment, would that be allowed? So there, various regulators have their own uh, rules or guidelines. So US allows interchangeability for few biologics, which the Europe doesn't allow, and depends on the various other studies which have been conducted. So then you may have to do a special study to ensure interchangeability of the two drugs. This is a NOR switch trial, so where you're able to switch from one to another. You need to prove that with all these data, and then you get a permission to switch. So as I was just mentioning, this is the reference product. This is a biosimilar A, this is a biosimilar B, and biosimilar C. So this is both way it's interchangeability. So you could either use A, B, or C. And then among the other generic available biosimilars A, B, or C, can you interchange between them? So that's known as interchangeability. The good part is that when we see in small molecules is like say after the patent expiry, there is definitely a dip in the cost of the drug. However, when it comes to biologics, there is no much of an impact even after the patent expiry, because those are typically considered as the different drugs altogether. There is an impact, but not that great. It depends on the competition per se. So if you consider erythropoietin, so when it was there marketed and the first one came, there was a gradual dip in the total market as well as the price, accessibility, everything was changed. When it comes to anti-TNF alpha type of a drug, there's no much of a change even entering into the market. So just summarizing about adalimumab. So it's been approved in the Humira, which is the reference drug, has been approved in 2002, and the other drugs from Amgen or Boehringer Ingelheim, they are typically biosimilar to the innovator of Abevir, that is Humira. On the and these is the same drugs which we saw the innovator as well as the other drugs from either say Amgen or Boehringer Ingelheim. So they get approval for all the indications if they are the innovator. However, if you are a Pfizer or a Amgen, you may not get in approval for other indications. Similarly, for bevacizumab, the innovator is Avastin in 2004, and then we have one from Amgen, and that's typically a biosimilar. So if we consider bevacizumab, it is the original one is indicated when all these different types of cancers, maybe small cell lung cancer, non-small cell lung cancer, or colorectal cancer, or glioblastoma, cervical cancer, renal cancer, ovarian cancer. These are all indicated. However, the generic or the follow-on biologics or biosimilar, you get approval for major of them, but you don't get approval for recurrent epithelial ovarian or fallopian tube cancers. Same the case with etanercept, the innovator from Amgen, followed on by biologics from Sandoz. As you could see, it takes nearly 18 years to get into the market, 1998 and 2016. So here you get approval in all the indications, psoriatic, ankylosing, plaque psoriasis, all. Infliximab, as you could see, the same year, about 18, 20 years to get into the market. And the Janssen was the innovator, Pfizer and Merck followed on. You get approval for almost all the indications except, say, pediatric ulcerative colitis. So, as I was just mentioning, we have these disease-related factors also which affect, and we come on to the endpoint factors. So, these are relevant to the disease state in the question. So, it could be how advanced the disease is would be affecting the efficacy of the formulation. So, there are some hard endpoints recommended by guidelines. So, which are sensitive enough to detect any difference. For example, rheumatoid arthritis, the evaluation of progression of joint damage by X-ray is not not required. So we have this infliximab. So these are various indications. The treatment methotrexate is allowed. What is a stable dose which is allowed? So that would be varying. So you need to have an endpoint which is hard enough, which should be able hard and sensitive enough to give you the difference between A and B. The another factor would be the sample size. So that is also important when it comes to biologics, because every patient counts, and the duration of the treatment should also be taken into consideration, as we've seen that some treatments are as small as 4 to 6 weeks, and some are as long as 24 to 52 weeks. So yeah, that's what we were seeing, the trial duration. So as we see, the as small as say 4 weeks, and there are certain as big as say 52 weeks also. The other factors which are important in case of the efficacy are the designing factors. So what we are targeting it, we are targeting it as equivalence or non-inferiority, or we are clinically putting it as a biosimilar sort of an design. So whatever you're trying to prove, you need a similar number of patients to prove your either equivalence or non-inferiority. So when it comes to non-inferiority, we demonstrate that it means that two products are not equivalent. I would say A is as good as B, but it's not A is B, right? So that's what we try to do is non-inferiority. The one is inferior to other, but it's still within the normal range which we have defined for non-inferiority. And when it comes to equivalence, these are most challenging aspects in study design, where you want to prove that A is equal to B, where you need probably stringent criteria to prove that A is equal to B. An example here, if we see, this is Remsima and Flecta. They did a study with 15% equivalence margin. So this is the margin which was considered, and the study was 30 weeks study. So these are the statistical parameters which were considered. And adalimumab biosimilar, which is recently approved in India, that is Exempia from Zydus. So they evaluated the clinical trial, which enrolled only 120 patients, and that was only for rheumatoid arthritis, and all patients were from India. So patient either received Humira or adalimumab of the sponsor. So there were only 60 patients being exposed, and the study was completed within 12 weeks. So the margin was considered when the innovator did the study. It was 15%, which is then made to 28.5%. So that decreases the number of patients you require for the study. However, with this large margin, you may be able to get an approval from India, but when you go to Europe or other US countries, they may scrap your study. You may never get an approval with this bigger margin. So there are various other statistical methods which are now used, which we call them as flexible approaches. So that brings me to one story, sort of like, say, we've seen the evolution of the mobile phones, right? So when, like, say from 2000 or we used to have those big phone sets, handsets from, say, Nokia. Hardly you able to text a message, or that was the basic thing. Now, with this in place, just a minute. Excuse me. Is it pronounced? हाँ बोला है उसको, वो कर रहा था. I did not talk it. What is the, वो फाइल बनाने के लिए बोला था. So that is on. हाँ मैं वो पूछ लेता हूँ. अभी एक इसका फोन आया था निखिल का, ये जरूर पे कॉल कर रहा हूँ, चल सकती वो काम कर रहा था. Yeah, sorry. So coming on to this adaptive design is what happens is, like, say, as we were talking, that we have mobile phones which have upgraded over a period of time when it comes to the previous phones and the current phones. However, when we come to our type of a setup where we get the same time to get the drug onto the market, as we've seen in the previous cases that were 16 years, 18 years to get the drug in the market. Now things have changed. I think if you have seen the vaccine getting into the market, so that was really quick. I mean, say people tend to believe that there should be some shortcut which has been taken into consideration uh, given the current situation, and the drug came into the market or the vaccine came into the market really quick, which is not the right case actually. Per se is the design, I mean, so the studies which were designed for this vaccine, they were adaptive designs or a seamless adaptive designs, sort of a new concept in designing, where you design the study such that you fail fast. So if you start, you fail, you scrap it. Again, you start, you fail, you scrap it. And so if you fail fast, you get a new option to try and treat. So that was what was used, and that's now what the new thing in adaptive design. So you change things based on what you are able to see. So if you're able to see some favorable response, you divert things onto that zone. Or you could say, if you see some sort of an efficacy, you do some interim analysis, you get some favorable result, then you add on more patient. Then you get into this a promising zone where you can get on to more information. But if it fails, then it's an unfavorable result and you scrap it. So that's how it goes. So that we call it as an adaptive design, and this is really advancing and helping get things done very fast and bring them quickly to the market. So just to get onto this adaptive design, you need a typical master protocol. A master protocol is one standard protocol which would be de-risking program that will be reducing the cost, the time involvement, and there'll be continuously there is a learning which is there, so which happens over a period of time, and definitely that has a huge advantage with respect to cost saving and the benefits with respect to the recruitment. So under the head of master protocol, we have two type of design. One is the basket trial or an umbrella trial. Now what happens is, let's say you have different diseases available. Like say, as we were seeing that there is a rheumatoid arthritis, plaque psoriasis, so on and so forth, right? So you have these multiple diseases which are there, and you try to treat them with one drug. Okay, so these are different diseases. You give this one drug, and that's the common targeted intervention given to all of them. Okay? On the other hand, what happens is there's a single disease. Okay, so if we consider, like say, there's a disease of breast cancer. Now, on the breast cancer, you target different drugs, like say drug A, drug B, drug C, and then if it works, that drug is targeted to multiple drugs are targeted to one particular indication, and the other way around is multiple indication, you have only one targeted drug. So, and there's a third one which is known as a platform trial, as we have the railway station. So something like that, that the train starts with all the passengers on board. When you try to test them with the standard of care, you give some drug, you give some other drug to these different patients, and you find that okay, this one is not working. So, and you do an interim analysis, you find that this one is not working, so you drop that arm. Those arm is dropped. You find something this is working, you add that arm. So now this arm was dropped, a new arm was added. New arm, you undergo interim analysis, you find that this arm as another arm you need to add, you add on another arm, and then you do a final analysis. So it's like a platform where people get down at their station and new people board in at the different stations, and you try to reach to a particular destination. These type of study designs or the master protocols is becoming more frequent and common now, and more studies are being designed with respect to these protocol design. These are few of the examples of master protocol. Uh, we have trastuzumab, vermafenib, erlotinib, crizotinib. These have been tried and tested through these master protocol. Because if you go by the indication of say just lung cancer, you may have scrapped the drug erlotinib or crizotinib. But when you came to know that this drug acts on this EGFR mutation and EGFR mutated or ALK mutated lung cancer, it shows a really good effect. Similarly, when trastuzumab was tried just in breast cancer, it would have been scrapped totally. And when we have this HER2 positive metastatic breast cancer, then it would be showing wonderful effect. So this is how the drugs were designed. I mean, say the trials were designed, and which gave a really good response. So they have a good advantage because the small sample size and the chances of hitting hit rate. I mean, say either you fail or you pass. That is really high because of small sample size. You just if it doesn't work, you just scrap that particular arm. The disadvantages are equally there, which is definitely challenging and a difficult study to uh, execute. So the single arm requires tumor response endpoint. So there is a high level of bar which has been set for getting the response. And similarly, overall, you end up with a longer duration, or you may end up with a larger sample size, considering all the sub trials which you have conducted. However, over a period of time, they have shown pretty much of advantages if we consider. So there have been a huge saving with respect to time as well as cost by using this master protocols. Just to share with you an idea, like previously where there were problems, I mean to say rheumatic patients, they used to receive drugs. If you could see here, the waiting time to get a drug was nearly 7.4 to 7.5 years, and then that decreased to zero point 0.7 years, zero point 0.5 years, and zero point 0.3 years, respectively, with the addition of Infliximab, Etanercept, and Adalimumab into the market. So there is an easy access or a faster access to patients of rheumatoid arthritis, which is being seen over a period of time. And we definitely know the cost that these are expensive medicines. But if you compare that of the penicillin, which was first discovered in 1941, and if you consider the cost of that in that particular time, it was nearly dollar 5,600. And with that, and that time of 1941, the cost of car was only 925 dollars, and the gold was nearly 33 35 dollars an ounce. So if you extrapolate this paracetamol, so penicillin cost in the current times, it would have been 7,000 7.5 lakh of dollars, and that would have been the cost in the current scenario. So these are definitely expensive, but we expect that over a period of time, the cost would definitely be reduced. Okay.